uh, how to determine the structure of a protein. Uh, from now on, we are going to see some of the basic techniques uh, which are required, which normally are used in order to determine, in order to uh, understand the structure of any protein. And of course, this is some of those techniques are simple and straightforward, and you know those techniques earlier because you might have learned about those techniques or might have used some of those techniques previously. But some of those techniques might be little bit new, and we will talk about them sequentially. Okay, so let's talk about uh, with this about this guy first. I mean, he's a very great name, very famous one, and I hope every one of you know this name, which is Fredrick Sanger. And you know what? Who was he? Fredrick Sanger. Some of you might know him because of uh, Sanger sequencing, because he is the guy who developed a technique for sequencing the DNA, and which later on was successfully used for sequencing lots of DNA molecules. Uh, he was a magic guy, a kind of, because he won 2 times Nobel Prize. I mean, this is one of the amazing thing which is very important about this guy. He won 2 Nobel Prize, one in 1958 and another in 1980. And he's a very genius guy, and he is one of the 2 people in the history who won 2 Nobel Prize in the same category. That is, for example, he won this Nobel Prize in chemistry. So this he's just one guy, and there is another guy who won this Nobel Prize twice in physics category. And of course, these are 2 genius guys. And if you just think what are the contributions of this guy in he was a British biochemist, by the way. He's a British biochemist, and he developed techniques for DNA, RNA, and protein. And we are going to learn some of the techniques, some of the things. I mean, we have already used those techniques developed by Sanger, but some of the techniques he developed for protein sequencing are also still in use, and they are very fundamental to our current understanding. Uh, he for the first time he was the one who in 1953 gave the structure of bovine serum insulin. Of course, it was not three-dimensional structure at that time, rather it was an amino acid sequence. First thing first, we will see in coming slides that the structural prediction cannot be possible without having the knowledge of primary protein sequence. So it was the first thing which you need to determine whether to what type of amino acids are there in a protein sequence. So he in 1953 for the first time he gave the sequence of bovine insulin, and of course this is the insulin molecule, very the same insulin which is also present in our body. But of course this insulin was not coming from human but from bovine. These are cattle, uh, our livestock. And he for the first time, and before that, by the way, before that, even before he started his work, it was believed that uh, it doesn't matter in what fashion the amino acids are linked to each other in a protein. I mean, the people were in generally believing that the amino acid composition is the decisive factor. It is the one thing which do matter only. So, for example, number of amino acids, for example, number of acidic amino acid or number of basic amino acid, or for example, prolines or histidines. The numbers of these amino acids, individual amino acids, do matters only. The the manner in which these amino acids are linked to each other, they do not matter. This is what people believed before that, but he for the first time showed that in fact the sequence of amino acids do matter. So not only the prolines individually, but the way how these prolines and might be any other amino acid, for example, histidine or any other. I mean, these are just the name. I'm just writing give you an idea what I want. So not the, uh, for example, lysine maybe as well. So it's not only the number of these amino acids which are important, but the way these amino acids are linked. They were also, this was the guy who for the first time gave us the idea, and of course today we do that. We believe that it is the similar. It is the thing which is which is the decisive factor. So, in addition to providing the molecular mechanisms of various proteins, information about proteins uh, structure give us a deeper understanding of evolutionary relationship between species. So that this is another two additional benefits about a protein structure. So the protein structure not only give us the insight about the molecular mechanisms, but also the evolutionary relationship. For example, if let's suppose there might be many proteins who have these three amino acids present in similar quantities, but it is rare that these amino acids are linked in the similar manner in more than one species or other. So this gives in fact the evolutionary relationship, which is also very important for this discussion as well. Uh, because some of the diseases, some inherited diseases are because of these protein structure. आपको पता होगा हमने molecular diseases का जिक्र किया था कुछ slides पहले, and these are so these these diseases which are inherited, they have something to do with the protein structure, and that is why the new diagnostic tools and clinical therapies uh, developed since then. The Fredrick Sanger has given us the first structure of the bovine serum. This technology, which was used in 1950, it required lots of resources. I mean, there were lots of people involved, lots of many teams involved, and of course, lots of resources involved. The technologies has been tremendously changed since 1950. For example, several groups worked about 10 years, about a decade, to give the structure of insulin, but this work can only be done nowadays, keeping in mind our advancement, technological advancement. A well-funded research team can do this whole job in just less than a 1 year. This is a very interesting thing. This is how we are doing nowadays. So, for example, we have lots of techniques. We love lots of our knowledge has expanded a lot, and we learn lots of things about the protein. So now we can do it much quicker uh, than we think it was possible before. And much of this thing has been uh, become easier because of automated technologies, and these automated technologies include, for example, the mass spectrometry, sequence determination, and blah blah blah. And because we have an automated sequence determination system, so the sequence Sanger did might be took maybe 5, 6, 7 years. It could be done now just within a few days. This is how it is happening nowadays. Okay, so a substantial portion of investigators' time is developed in extraction and purification. Even today, if you just talk about the protein structure determination, if you want to do that thing, so you see much of our efforts, much of our resources, and much of our uh, strategy is focused on how you are going to isolate your protein of interest and how you are going to purify it. Because once you purify it, you can go for amino acid sequencing, you can go for structural prediction. But if you do not have purified protein, everything is just a dream. I mean, you can think, you can dream it again and again, but you cannot do it. So the first step, extraction, and then purification. And of course, these are not so trivial things. There are two fundamental problems which are associated with this step of the protein structure prediction. And these two fundamental things are, for example, there are low amounts of protein. When you talk about a cell, so there are thousands of different proteins, roughly about 10,000 proteins we have in our body, in our cell. So that's roughly about 10,000 proteins. And if you just think for a moment, what does that mean? So we have a cell which is roughly about very small in size, maybe uh, as we said, about 10 microns, or maybe 10 to 100 microns. And in this 10 to 100 microns, you have proteins, and there are 10,000 proteins. And if you think this is a small compartment, in this small compartment, there are very minute amounts of proteins present. And of course, when it comes to isolate, how can you separate this? It's just like a desert, and in that desert there are some small salt grains, and you are going to separate these. You are going to pick those salt grains individually, and this is how you, what you mean about low amounts. So this is a protein is extremely small amounts present in the body, uh, and it's not only that there are small amounts of protein. Out of these 10,000 proteins, you might be interested in only one of those proteins. So this is even a black cat somehow caught in a dark box, and which is put in for maybe in a dark night, and you have to find out where is this cat. So this is very difficult thing, that there is a problem above the problem. So you have very low amounts of protein, and of course not only one protein is in low amount, there are multiple other proteins which are in low amounts, and out of those 10,000 proteins you have a challenge to separate this. So you need to get an idea, you need to get a technique which could overcome this thing, which could overcome this problem. So this is the low amounts. And then there is an optimal condition. The second thing, it's not only the uh, amounts which are important when we talk about structural determination. The conditions in which these proteins are working, they are very important. Now all the proteins have specific conditions. For example, they have specific pH to be alive, they have specific temperature to be working fine, they have specific cell concentration where they are much better than other salt concentrations. So this, of course, these factors are very important. So when you talk about protein, it's not simply taking those proteins out from the cell and then let them know what they are exactly. And in order to do that, you have to have all these parameters fixed before doing all these things. So this is a two critical steps: low amounts in optimal conditions, and because of that, still our major portion of the time is investigated, is somehow dedicated to extraction and purification technology. We will talk about them extraction and purification technologies shortly. Uh, there is just an example given here. I mean, it will just tell you how these two things are very important. For example, the problems in handling protein may become apparent only after considerable time effort. So example is given. For example, the investigation of nitrogenase, which is an enzyme, which of course is a very important enzyme in environment, and this enzyme, what this enzyme do, it reduce N2 to form an NH3. So this is a very important enzyme. If you just do not have this enzyme in the plants, in leguminous plants and in roots, you might not be able to fix this nitrogen and made it available for the plants to make proteins and other stuff. What was the problem in effect? The problem was we did not know that oxygen exposure could destroy this. So you see, this is a very simple thing which took maybe 1 decade to figure out how important it is not to take this protein out of the cell and expose it to the environment, because once it is exposed to environment, it is destroyed. You cannot do any work to that. So this is very important. So that is why when we talk about optimal conditions, we have to know those conditions in which this protein. So you might be able to separate nitrogenase in big quantities now from the cell. So you took maybe one truck of this leguminous plant roots, and then you crush it, and then you purify it, you have that protein. But still you are not able to go for the protein structure because you haven't got the optimum conditions for that. We will talk about them. So there are some certain steps which we are going to talk one by one in coming slides, and of course this is going to be somehow an interesting lecture, or maybe a little bit longer lecture. So first thing first, you have to isolate the proteins, and then you have to purify them, and then of course second step is to know the amino acid composition, what type of amino acids and in what quantities, and at what percentages those amino acids are present in those uh, proteins. And of course uh, degradation of proteins or polypeptide into smaller fragments. So if you just divide it, how it is going to be in smaller fragments, and what is the sequence of this protein. And then of course there is a fifth point which is not written here. It's the three-dimensional structure, and for that I will bring it in a separate lecture. You can go for NMR structure, X-ray crystallography. The in silico resources are there to predict the structure. So first you have to know all these things, and then you can go move forward to those things. Uh, Proteins vary in size, charge, and water solubility. So different proteins have different these characteristics, and no single method can in fact work for all these categories. So there must be proteins which could be separated through on the basis of size, but not on the basis of charge. But there are proteins could be separated, might be useful to be separate on the basis of charge, because size do not work for there. Or there might be proteins which might be simply separatable on the basis of solubility, their solubility properties. So there are different. So we have roughly 10,000 proteins. I mean, this is a huge number of proteins, and of course, these are those proteins which we know today. There might be other proteins which we still do not know, and of course, we are still working on a much better pace than 100 years before, and now we are doing it much quicker than it was doing before. So it's a huge task to separate proteins, and uh, and out of these all proteins, so what will you do? So you took all these 10,000 proteins first. You cannot afford to lose any of those proteins. So think first what I'm saying. So you want to work with a protein, and you have a protein A, and in order to separate protein A, it's not easy. It's not like that. It's not a magic that you just take your hand out and take protein A. For that, you have to take all the proteins out of the cell. So these are from A to Z, all proteins. Now you cannot miss any of them, because you do not know whatever you are missing might be the protein you are interested in. So you have taken all these proteins out. So first thing, you are taking all those proteins, separating all the proteins, and then you are looking for that specific protein out of these 10,000 proteins with which you are working. So this is another thing uh, which is important. Uh, any molecule, whether protein, carbohydrate, nucleus, can be separated or resolved from each other on the basis of physical characteristics, physical or chemical characteristics. I have already given you those physical and chemical characteristics on which you can separately separate them. When you talk about proteins by perspective, there are two very important physical characteristics. I haven't highlighted it, and I should have highlighted, but I forgot. Maybe somehow let me. So these are two very important factors when we talk about proteins. These are size and its binding affinities, which are very important when we talk about protein purification. So you have now 10,000 proteins, and you could separate them on the basis of say size and binding affinities. Of course, there are other factors, but most of the time we are mostly doing it through these techniques, size and binding affinity. So two most widely used characteristics are separating them on the basis of size and on the basis of binding affinity. So we are going to see both of them in this section, isolation and purification section, um, because these are very important in a way. This give us a mean to work to start with something. So there are we know It's not. It's impossible that all the proteins could have the same size. Why? Because they all the proteins have different amino acids number. So of course they have different amino acid numbers. They have different size. So you could make a plan to separate larger protein from smaller protein, or from the very smaller protein. Or you could also devise a way that a protein which could be able to bind, for example, to a specific resin, for example, a charged resin. For you know your protein might be positively charged, so you could be interested to make make a make a pot make a pot which is negatively charged all the way, so that any protein which is positively charged will bind it here. And of course, we are not sure that this protein will be of your interest, but we are pretty sure if all the proteins in the cell who have positively charged, they are present, they could be hold in this container, your protein will be one of them. So this is how we do normally things. So you have a characteristics. For example, you have a binding affinity, and you take this binding affinity to separate those proteins. So this is how. So one of the techniques to isolate proteins is the centrifugation. Centrifugation can separate particles on the basis of their size, molecular mass. Of course, all of you know what is centrifugation. Principle is very simple. The first thing, I mean, of course, normally it is always the first step in a typical protein purification scheme. So you start from anywhere, you will go for centrifugation first. You start from any material and you will do it the same. It's not the rule. It's not a rule. ऐसा नहीं है कि ये कोई कानून है, but it's just the most suitable thing we do. So we start with centrifugation so that we can separate things. So you have a cell, आपके पास cell है, cell के अंदर मुख्तलिफ चीज़ें हैं, और आप चाहते हैं कि उनमें से आप अपनी पसंदीदा चीज़ को निकालें, तो उसके लिए आप सबसे पहले centrifugation करें. So this is how we do. So centrifugation can separate parts. So what is the principle? Principle is two particles in suspension, which could be anything, cells, organelles, molecules, but the condition is they must have different mass densities. They should settle down on different rates. This is the principle. So any two things which are different from each other by their mass or by their density, they could be separated. This is the very simple principle of centrifugation. So that's also very alarming, because if we are interested in two fragments are maybe which are having the same mass, you cannot, you see, are the two proteins which have the similar densities, you can separate it centrifugation. So this is simple. So no, the wave density is the ratio to mass volume, ratio to weight volume, ratio to weight to volume ratio. It's a weight to volume ratio which is called density, and there is a chart which gives you all these categories. You can separately calculate volume from using mass and density. You can calculate density from, I mean, this is just to give you an information. It has nothing to do with our current scheme, but it is important to understand that centrifugation work either on mass or on density. So more denser materials could settle down quicker, and more massive materials could settle down quicker, and this other thing. Proteins vary greatly in mass but not in densities. This is an important thing. Why it is important to understand? We know that most of the proteins have different mass, but of course their density is not different. Why it is so? It's just because proteins are small molecules, and of course when doing the folding stuff, their volume, overall volume could be changed. So it could be possible that this change in volume affect the density in this. So by the end of the day, the density remains a little factor for most of the time for protein. So that is why most of the time when you talk about proteins, you talk about their mass, because their densities in a solution in a liter. So you have all the time 1 liter So it's a very little, very delicate change which could be measured in density. And of course, it is much easier to measure in terms of mass. So that is why we mostly do in terms of mass. So until unless some proteins are attached to other molecules, for example, carbohydrates or lipids, their densities do not change. So their densities most of the time remain the same. And now because we have attached to other molecules which are bigger in size. For example, we have lipid molecules which are bigger in much bigger in size. Carbohydrates much heavier and so that could lead to the change in densities, but normally they are not. So most of the time proteins have an average. For example, you can say density of 1.37 grams. So that means it's similar. It's very small amount. So for example, could change to 1.39 or to 1.27, but it's not a big difference. So that we could imagine we could we could separate it, and these are within not more than 15% change. So that means 15% of 1.37%, if you just calculate it for the sake of understanding what I mean to say, 15% of 1.37 gram might be 0.001 gram per centimeter cube, which is such a small number, which is such a small number, which is not possible somehow to measure through centrifugation. So that is why we do not think of that time. Heavier or more denser molecules settle or sediments more quicker than lighter or denser molecules. We have already talked about that quickly. So we know now the principle. What is the principle of centrifugation? We know that the more denser molecules, more quicker molecules could be separated out on a different pace. So what we do normally? So we take normally your sample, whatever is that sample. You have a protein, not only proteins. Of course, it's a sample of cellular extracts, which will also contain some DNA, some RNA, some carbohydrates, some proteins. So you have a protein, and you centrifuge that in a centrifuge machine. And what does that mean? It gives a centrifugal force, and centrifugal force is the force which pulls things to outer side. So it will pull you to the outer side, and you have a rotor down here to which this is attached. So greater the molecule, more further it will go down. So for example, these are three different molecules which will go to this centrifugal force. And as greater centrifugal force, as million times. So this is one. I mean, it's a huge number. Force of gravity. So this is not RPM only. This is gravity. So you think for a moment. What is G? G is one thing which pulls us to the surface, to the Earth, and 1 million times greater centrifugal force could be applied in a centrifuge machine to separate these things. And this force is so huge that it could separate out a molecule of the size of 10 kilodalton. It's a small molecule. It's not a big molecule. It's a very small molecule. Sometimes it is a very hard to see it on the gel. So this is a small molecule. But by centrifugation, putting this centrifugal force, which is huge centrifugal force, and pulling, you can separate 10 kilodalton. Even still, proteins which are smaller than 10 kilodalton cannot be separated through centrifugal force. So centrifuge. So this is, you see, this is another setback. This is another thing. Says only 5 kilodalton proteins with a say 10 kilodalton could be separated. Modern ultracentrifuge even could go beyond that limit, and we could maybe run for 150,000 revolutions per minute. These are called RPM. So this is a huge 150,000 revolutions per minute. So they are spinning so quickly that it produces a large amount of force which could separate smaller molecules. But even still, 5 kilodalton molecules is not possible to to do. Centrifugation normally is used for two types of purposes. So this is given here. So one either is a preparative technique, either is an analytical technique. So I believe all of you are familiar with these two different things. Preparative technique is something which is to prepare for an experiment. And analytical technique is something which is to analyze an experiment. So these are two different things. So let me give you a quick overview of these two things. Preparative technique might be that you are preparing the sample for some particular experiment. So this is preparative. For example, you are going to do a PCR machine, PCR reaction. Your experiment is PCR, but for doing PCR, you have to prepare the sample. And what is that preparation? You have to separate the DNA in big quantities. So this is called preparative. So you are preparing the sample. So similar is the case with the protein. For example, you want to do SDS-PAGE, you wanted to do NMR structure prediction, or you wanted to do any other thing. You have to prepare the sample, and that preparation means you are doing it on a larger scale. So it's not now one cell, it could be millions of cells or 10 millions of cells, greater cell material, so that you could get more amounts of protein. So because you are working with 10,000 proteins, and out of 10,000 proteins, it's very hard to separate one molecule. So you have to increase the number. So that is why you might not be working with 10,000, you might be working with 1 million cells. So because in 1 million cell, there are much better chances that you might have 100 molecules present. You might have 1,000 molecules present. And if you work with maybe 1,000 proteins only, 1,000, uh, let's for a moment, if you are working with 1,000 cells only, so in 1,000 cells, how many proteins you are going to separate? 10,000 proteins. Think, 1,000 cells, 10,000 proteins. So might be your protein fraction might be 0.00001%, which is even close to impossible. So that is why you do preparative technique. So you prepare on a bigger scale. And then there's an analytical thing. You actually do an analysis to measure physical properties, for example, the molecular weight, density, shape, equilibrium, binding constants of a micromolecule. So, and for both purposes, you could go for centrifugation stuff. I haven't. I have just given you a chart here. It's not important. It's just to give you a feeling that not RPM means not always RPM means the G value. The G value is calculated on the basis of RPM, and of course, this has to do with the RCF, which is a relative centrifugal force, which is applied to this. So you should know the rotor size. What is the radius of your rotors? You should know the RCF value, and you can give the RPM value in this formula, and then you can calculate the centrifugal force and everything, which is important. So this is the similar thing. Okay, now let's quickly move to centrifugation thing. So this is centrifugation. This is simple centrifugation. There are two types of centrifugations we normally do in the laboratory. Of course, there are other centrifugation. We are not working. We are not interested in them. So one is the differential centrifugation. Of course, the name said it could differentiate between two things. So either you are interested in soluble fraction or non-soluble fraction. So this will differentiate both of them. So might be you are interested in a preparation and you know, if you have the idea, if you have the knowledge that your protein of interest is soluble fraction cytoplasmic, so you could go for differential centrifugation. It will separate out all the cytoplasmic material and will separate the nuclear material separate. So this is centrifugal differential centrifugation. So it is happening now, differential centrifugation. So soluble proteins will be in the supernet and insoluble proteins will be in the bottom. So how you do that? You have a cell homogenate, for example, a cell extract, total cell extract. You didn't do any specific thing. You took the cell. It could be cancer cells. They could be plant material. They could be a root material. Any other thing. Any fungus. Any yeast material. You crush that cell. You grind them. You put them in some alkali solution to break their cell wall, cell membrane. You have the cell homogenate, and you just pour this in a tube, and then you spin it on a much longer, much larger speed, on a rotor speed, the maximum rotor speed. That means maximum rotor speed. And what will happen? You will separate out the nuclei. For example, this is the suspension now. So there are denser particles, there are less denser particles. You centrifuge it, it will spin this quickly. You have something in the supernatant, and the other things are settled down in the form of pellet. A pellet is down there. So you could separate both of these fraction in two different tubes. So you have taken that supernatant into a separate tube, and then yeah. So this is the differential centrifugation, and what is the outcome? If your protein of interest is present in the soluble fraction of the cellular extract, you could go for that. I mean, you could go further experiment. You could take this and you could perform further experiments, and you could go maybe for example, gel electrophoresis. Okay, or you could go for maybe uh, go for emissive spectrum or whatever you want to do. It's not important here. So you can do, and if you know your protein are in the nuclear fraction, you have to further digest that. So you will digest it, you will further do, and then you may come back but do another centrifugation again. This will be the differential centrifugation, because some of them will be in the supernatant, some will be settled down. So you see, you can sequentially do centrifugation one after the other, in order to take your protein of interest out of that. So this is just a normal centrifuge machine present in any lab. You might have seen. Normally, it do not run more than, I mean, normal centrifuges machine which is present in our lab, 10,000 to 15,000 is the maximum RPM speed. And of course, this RPM speed means uh, nothing when you talk about proteins. This is a very trivial thing. So that means one thing: you cannot use the normal tabletop machines for protein centrifugation stuff. Of course, differential centrifugation could be done through this, because these proteins, if they are present in cytoplasm, they will be present in this fraction, and of course you can separate them out. Then there is another centrifugation method which is called rate zonal centrifugation method. You might be familiar with the density gradient centrifugation tech thing. It is a similar thing. So this is working in a almost in a similar fashion, but with slight differences. How these differences are that instead of doing uh, making the protein on a normal uh, mixture cellular extract, you load the protein. So what you did in previous slide was you took the cellular fraction, you didn't make any specific material to hold this thing up, to hold this cellular extract. So that means there was no specific buffer made for centrifugation, there were no specific material made for centrifugation. But in rate zonal centrifugation method, you create an artificial density gradient in which the proteins of interest could be separated on the basis of their densities, on the basis of their molecular weight. And how you do that? You make a specific mixture of protein, specific material, uh, which once when spin, it will create a gradient of, for example, heavier, less heavier, much heavier. That is why it, this technique is also called a density gradient technique. So you have much better option to separate different mass. Instead of, there are only two options: supernatant. Previously, normal differential centrifugation, there were two options: either the protein is present uh, in the supernatant or in the bottom, either you are with us or with not us. But here you have much better option. You could separate proteins on a much bigger. So that is why, so what will happen by the end of the day, you will be ending up with different discs of protein. So this is not one only. So there are multiple bands appearing in the tube. So each of these bands have different density or different molecular weight. So that is why it is called density gradient. You could say them zonal material. So that is why it is called rate zonal. So this is a different disk. This is a different zonal disk. This is a different zonal disk. So each of these zonal disks are based on some density gradient in which they are working. So how you normally do that. So a protein mixture is laid on the top of a sucrose gradient. So most probably, most commonly we use sucrose. It's easy to prepare, it's easy to make. So you make a concentrated solution of sucrose, maybe 10 molar solution is sufficient, sometimes 20, it depends what you want to do. So 10 molar solution, for example, of sucrose once made and spin on a high speed in a centrifuge like this. This is not a normal centrifuge machine present in the lab. It's a floor centrifuge machine which is bigger, just like a refrigerator or washing machine in your home. Of course, your washing machine in your home is also a centrifuge machine. If you think for a moment, what it do is the same thing. You have a rotor which spins quickly. Think so. Anyways, it's much different than that washing machine. So it's a ultra centrifuge machine. You have a rotor, and this rotor could go on a much higher speed. For example, just मैंने बताया पहले 1 million G तक लेके जा सकता है, even more than that. You will go for that as well. So you took this sucrose gradient in the tube, and in this sucrose gradient you could go for ultra centrifugation. The mixture will be this sucrose gradient will prepare the density gradient, and of course all the proteins which are present in this mixture will be start separating from top to bottom on different bands in a different disk, zonal disk, on the basis of their mass, and you could separate them. So you see, this is the depiction of how it will happen. So you have a sucrose gradient, this blue color, and then you loaded your protein fraction up there and centrifuged it on a quite high speed. So you see there are proteins heavier, there are proteins less heavier, and there are proteins even at the bottom. You could separate all these fractions in different smaller products, larger, small, larger proteins and largest protein, and whatever they are, you could separate them all together. So this is called rate zonal centrifugation stuff. But you have to be cautious. There are two things which where you have to be careful when you talk about this centrifugation, which is called delta centrifugation. What is that? If you spin this mixture for a very short period of time, you will not separate anything. You see, I mean, in a normal centrifugation step in the lab, we do normally centrifugation maybe for 10 minutes, 20 minutes, maximum 30 minutes or 1 hour. This is a short time for ultracentrifugation normally. So you have to spin it sufficiently. Maybe from 5 hours, 6 hours to 12 hours is sufficient. It depends on what type of molecule you are interested. But at the same time, if you think you are going to do it on a longer than necessary time, for example, your optimum time was 12 hours and you spin it maybe for 15 hours, you may end up everything in the pellet. It could be possible everything will be pelleted. So what does that so, I mean, that means longer you put a pressure on this smaller molecule, which is under my cursor at this time, this way, this one. If you even put the pressure for longer period of time here, even this could be settled down. You see, this is a very important point. You understand. You need to understand what is your basic purpose. The purpose is to separate them, and it's not the purpose that you want by the end of the day going to join them. So you have to avoid this fate, and you could avoid it if you do not run it for longer than necessary time period. So this is rate zonal centrifugation, and I hope you have now the better idea what are the two differential and rate zonal centrifugation. Although Sometimes rate zonal centrifugation is seldom effective in determining precise molecular weights. ये आपको जरूर याद रखना चाहिए. You cannot get a precise thing here. You will get an idea, for example, the molecules from 100 kilodalton. Sorry, मैंने गलत मैं लिख देता हूँ. Molecules from 100 to maybe 10,000 size. So you see, this is not the exact size. Similarly, the centrifugation, any type of centrifugation, could be affected by the shape of molecule. This is an important thing which you need to understand, that even smaller molecules, if could take a much better shape, they could act like a heavier thing. They could act like a heavier thing. एग्जाम्पल मैं आपको दे देता हूँ, आम जिंदगी से एग्जाम्पल देता हूँ, आप बाकी चीजें तो आप देखते रहते हैं. दिस पेपर much high, much lighter, simple, two-dimensional. If you just throw it like this, it do not have any weight. It could fall anywhere. But if you wrap it, crush it like this, it's much heavier now, and it have a specific fate now. It can go to a particular direction because under the force, under a particular force. So that means even if there are smaller protein molecules, they could become heavier because of their shape. So that is why you cannot rule out this phenomena of the shape. So the shape could affect the sedimentation. So again, you will get a rough estimate. You will not get a pinpoint estimate. So that is why whenever we do experimentation, electrophoresis, we are always concerned whether it's a double stranded molecule, it's a single stranded molecule, and of course both of them are important. अगर आप लोगों ने इस यहाँ हमने टू डायमेंशनल जनरल इलेक्ट्रोफोरेसिस देखी, सॉरी centrifugations देखी. If you wanted to ask anything, you could go on. Any questions until now? मैं कब से बोल रहा हूँ, कुछ आप लोग भी बोल लीजिए.
सर जो हमने पहले पढ़ा कि डिफरेंशियल जो हमारे पास सेंट्रिफिकेशन है और ये जो दूसरी रेट जोनल सेंट्रिफिकेशन है, सर रेट जोनल सेंट्रिफिकेशन में हम ज्यादा प्रिसाइज जो होंगे एटम्स उनको सेपरेट कर सकते हैं.
Yes, exactly. The rate zonal is much more precise than the differential centrifugation, because it's in differential centrifugation you will end up with two separate phases. Either it is a soluble phase or it is a pelleted phase, and this will be mixture of many things. But in rate zonal you will get multiple discs, and of course you could have a much better approximation of your protein of interest. For example, if you are working with a protein which you believe is of the size of, let's suppose, 700 kilodalton or 100 kilodalton, so you could go for a particular zone where that protein is present. So you can separate that protein out. So for example, you might be more precise that you have to think of this fraction further. So अब आप इस fraction को मजीद देख सकते हैं further experimentation के लिए, and you could rule out further. So it's a more precise than this one. ठीक है
सर जिस तरह सर एंटीबॉडीज के जो डेंसिटी है वो सेम हो सकती है, लेकिन उनके मैस डिफरेंट होते हैं, तो ये मैस की बेस के ऊपर सेंट्रिफिकेशन ज्यादा प्रिसाइज मेरे ख्याल में जी सर कर सकेंगे.
यस, अभी इसके बाद हम जो टेक्निक देखने वाले हैं जेल इलेक्ट्रोफोरेसिस, उसमें हम दो किस्म की जेल इलेक्ट्रोफोरेसिस देखेंगे, उसमें भी हम डिस्कस करेंगे. Once you do, sometime you are better approximation on the basis of mass. आप mass को separate कर लेते हैं, लेकिन it could be possible two proteins have the same molecular mass, जैसा कि बहुत से cases में होता है. तो उस केस में हमारे लिए मुश्किल हो जाता है कि how we could determine, we could isolate that protein. So for that purpose you could go for other characteristics. Normally a rule of thumb is you do one technique and then you do the second technique, and then you do the third technique. So for example, you did the centrifugation, then you did the gel electrophoresis, and then you might be able to take this electrophoretic stuff taken out and then go for another technique, maybe the mass spectrometry. And so this is a sequential stuff, so one technique to the other. So mass is of course a much better approximation than density. So you are right, because हमने भी पिछली स्लाइड्स में देखा कि डेंसिटीज का इतना फर्क नहीं पड़ता प्रोटीन्स के ऊपर जी. No. Okay, now let's move on to the next. I don't know how much we have time left, so it's 5 o'clock. Okay, let's quickly move to the next slide so that we could ज़्यादा नहीं, सिर्फ एक दो स्लाइड्स और देखेंगे और उसके बाद फिर हम कल करेंगे. So we have seen about the centrifugation, but there is another technique which could separate things, separate protein, and this is called the electrophoresis. And if we all know what is electrophoresis, the name says under the influence of electric current, you are going to separate molecules from each other. And most of the time, electrophoresis work on charge to mass ratio. It's a very, very fundamental thing. It's a charge to mass ratio, which normally electrophoresis is working. So you have an electric field. You prepare a gel. A gel is any matrix. Gel is anything, any container, any holder. A gel हमारे जहन में शायद है क्योंकि solid material ही होता है. It could be a liquid material. It could be any resin. It's a gel like thing. So this you create an electric field in there. You have a high voltage, and then you put a buffer. And because buffer and electric current, there will be two possible scenarios. There will be an anode thing, and there will be a cathode thing, and there will be molecules which either move from which will prefer to stay to this anode portion, and there will be molecules who love this cathode portion. So those molecules who love this cathode portion, they will try to move quickly, and of course this will be affected by something which is called mass. Now one thing, it could be possible all these proteins have one negative charge. So if all these proteins have one negative charge, so there is no preference for the cathode. So what is the other factor which could be hampering this whole thing, which is their mass? So that means you could separate proteins on the basis of let me take this one. Oh, I wanted to take a resin. So you could separate things. You could separate proteins on the basis of mass to charge ratio, charge to mass ratio. And if all the proteins have the same charge, so of course the decisive factor is there, the mass. So heavier molecule proteins will be moving much more quicker than the lighter proteins. So electrophoresis is a technique for separating molecules in a mixture under the influence of an applied electric field. हमने पर electric field का जिक्र किया. Sorry, dissolved molecules in an electric field move or migrate at a speed determined by their charge to mass ratio. अभी मैंने जिक्र किया charge to mass ratio को और समझाने की कोशिश की कि कैसे एक वक्त के ऊपर चार्ज बिल्कुल बेमानी हो जाता है, क्योंकि तमाम अभी हम आगे जिक्र करेंगे जब हम इस टेक्निक को थोड़ा सा फर्दर देखेंगे. When you do this technique, you normally treat proteins with SDS, and this SDS give maximum charge to all proteins. So no matter whether the protein was positively charged or negatively charged, they all have the same charge now. So that is why the charge does not matter anymore, and now this is the mass who is playing a much better role. So for example, if two molecules have the same mass and shape, the one with better charge could be separated out. And of course, this mass to charge ratio now will play a very vital role in those proteins which have only the mass factor. So this is how it is happening. So, oh, sorry, I just forgot to. So I wanted to play this thing, why it is not going to be. So when you do electrophoresis, something like this happens. So the larger proteins move slower, medium proteins move faster, and the smallest proteins could move even much faster than this thing. And this just because of the electric current which is applied on this scheme, and this is how it is happening. So in SDS-PAGE gel electrophoresis, it is called SDS polyacrylamide gel electrophoresis. It is just the technique. We know some of you have used it already in the lab for their research purposes. Some are not familiar with that, but of course it's not that much alien. It's somehow useful for all of us. So SDS polyacrylamide gel electrophoresis. You made the gel in a pair of glass plates. So you have a pair of glass plates like this, and you pour the protein fraction, and you pour the gel mixture in this glass plates, and this is the polymerizing solution of acrylamide. So acrylamide is the molecule which is going to be polymerized and then cross-link and then make a semi-solid matrix, a gel-like stuff. The pore size of this gel are important because they will decide how quicker the proteins will move. So if the pore size is bigger, almost no protein will be separated because all the proteins can easily pass through this big hole. जैसे अपने एक दरवाज़ा है, अब दरवाजे को एक सुराख कंसीडर करें, उसमें से हर बंदा गुजर सकता है, लेकिन अगर आप इस दरवाजे को छोटा कर दें तो छोटे साइज के जानवर ही गुजर सकेंगे। अगर आप इसको एक छोटा सा कर दें तो बिल्ली गुजर सकेगी, और उससे भी छोटा कर दें तो सिर्फ चूहे गुजर सकेंगे, उससे भी छोटा बना दें जो कि आपको नजर नहीं आता तो शायद उसमें चींटियां गुजर सके। दिस इज द पोर साइज विच इज इम्पोर्टेंट। सो ऐसा ना हो कि आपने पोर साइज इतना बड़ा कर दिया कि आप कोई प्रोटीन सेपरेट ही नहीं कर सकते। सो दैट मीन्स everything will be at the bottom. But if you have a smaller pore size, very smaller pore size, that means you can only separate small proteins. So larger proteins cannot be separated because all the larger proteins are up there. आप इसको एक लम्हे के लिए सोचिए, मैं आपको उर्दू में बता रहा हूँ, एक लम्हे के लिए सोचिए आपके पास प्रोटीन्स हैं 1 से 100 नंबर तक, from 1 to 100 numbers. So if you make a pore size, if you make a pore size of, for example, 2 micrometer, what will happen? Only the proteins with 1 micrometer will pass. Where are the rest of the proteins? They are all up there. They are not, they haven't even in the center. They are all up there. And if you have a pore size of 100 micrometer, what does that mean? So you have all proteins down there. Nothing is up there, nothing in the center. So what does that mean? It's a very important thing which you need to understand, that you have to decide the pore size very intently. So you might be interested, again, it's your perspective. You might be interested in protein fractions from 1 to 10. So you will try to choose a scale which could separate proteins of 1 to 10. So that means you might be able to make a pore size of 10 micrometer by changing the concentration of this acrylamide. So now you can change. So इसको यूज समझिएगा, अगर आप 100% acrylamide यूज करेंगे तो pore size 0. So nothing can pass through it. If you check the pore size, if you use 90% acrylamide, there will be pore size, smaller pore size, maybe 10% proteins could pass through this. If you use, for example, 80% acrylamide, that means you are making a pore size for 20% of the protein. If you are making 70% acrylamide solution, you are even increasing the pore size. So lesser the material, more big pore size you are going to need. More the material, much smaller pore size. So it's inversely proportional to the pore size. So the concentration is inversely proportional. Okay, so concentration is inversely proportional to pore size. Let me write it like this. मुझे आहिस्ता आहिस्ता लिखना आ ही जाएगा आप लोगों को पढ़ाते पढ़ाते. So the pore size is inversely proportional to. Oh, देखा मैंने उल्टा लिख दिया, आप लोगों को समझ नहीं आया. Let me remove it. मैं अच्छा बच्चा बन के सीधे तरीके से लिखता हूँ. So pore size is inversely proportional to this acrylamide concentration. So you decide the pore size more and carefully so that you can separate the protein. I am not going to आप इसके मतलब खुद से पढ़िए बहुत ज़्यादा, इसके ऊपर मज़ीद फोकस नहीं करने वाला. Before loading onto the protein, proteins are exposed to the ionic detergent which is called SDS. मैंने आपको जैसे बताया पहले भी, SDS stands for sodium dodecyl sulfate, and this sodium dodecyl sulfate denature the proteins. That means any protein, any secondary structure, any tertiary structure is no more there. So proteins secondary tertiary structures are gone, their hydrogen bonds are gone, and now the proteins are just one dimensional molecule on the basis of size or size. And more second thing which SDS do, it load a lots of negative charge on the protein. So no matter whether the protein was positively charged or neutral, all the proteins have now the same charge because it's excess of so it's SDS. So this minimize the effects posed because of shape on migration. So अब जैसे मैंने बताया कि अगर शक्ल चेंज कर देंगे तो इसकी इनके चलने पे फर्क पड़ेगा, इनकी माइग्रेशन पे फर्क पड़ेगा. बस मैं बेसिकली थक चुका हूँ इस वक्त. Even chains that differ in molecular weight less than 10% could be separated out. This is just, let's go to this picture. So you see these are proteins. आपके पास बहुत से proteins है, smaller भी हैं, heavier protein भी. SDS से साथ कोट कर दिया, इसको इस जेल के अंदर लोड कर दिया, इसके अंदर इलेक्ट्रिक करंट लोड किया. आपको पता चलेगा कि इलेक्ट्रिक करंट तो जो हैवियर प्रोटीन्स थी वो, जो स्मॉलर प्रोटीन्स थी वो ज़्यादा इजीली इन पोर साइज के साथ गुजर गई, जो हैवियर प्रोटीन्स थी वो नहीं. इसको थोड़ा सा सेंट्रीफ्यूगेशन से सेपरेट कीजिएगा. आपके जहन में एक थोड़ा सा खदशा हो सकता है, सेंट्रीफ्यूगेशन में हैवियर चीज़ें नीचे को जा रही थी. यहाँ पर क्योंकि हमने कंस्ट्रेंट साइज का लगा दिया है, पोर साइज, तो यहाँ पर लाइटर मॉलिक्यूल्स नीचे को ज़्यादा आएंगे, हैवियर मॉलिक्यूल्स नहीं जाएंगे. ठीक है जी, मैं इससे ज़्यादा नहीं आज बोल सकूँगा. मजीद कोई क्वेश्चन है तो प्लीज़ काइंडली आप कर सकते हैं. जी क्वेश्चन.
सर
जी जी
सर इससे थोड़ा सा रिलेटेड था, तो जिस तरह आपने बताया कि
जी जी
प्रोटीन्स के मैसेज भी सेम हो सकती हैं, उनके डेंसिटी भी सेम हो सकती है सर. कुछ या तो इनको कंबाइन
आपकी आवाज मुझे सही सुनाई नहीं दे रही, कट
कुछ ऐसा मेथड हो जो प्रोटीन्स को
आप अपना क्वेश्चन दोहरा दीजिए. मुझे
केमिस्ट्री के अंदर सर मैंने ये पूछना था कि जिस तरह केमिस्ट्री के अंदर एटम्स की बेस, एटॉमिक नंबर की बेस के ऊपर पीरियोडिक टेबल बनाया है, क्या हम प्रोटीन केमिस्ट्री में प्रोटीन का पीरियोडिक टेबल बना सकते हैं?
आप कह रहे हैं प्रोटीन का पीरियोडिक टेबल ऑन द बेसिस ऑफ चार्ज टू मास रेशियो. देखें, असल में प्रॉब्लम यह है कि वहाँ पर पीरियोडिक टेबल के लिए एक ख़ास खूबी इस्तेमाल की गई थी, जिसको हम मोलेकुलर मास कह लें या इलेक्ट्रॉन, नंबर ऑफ इलेक्ट्रॉन्स जो थे उनके अंदर, उसकी बेस पर पीरियोडिक टेबल. देयर इज नो सच कैरेक्टरिस्टिक्स present for the protein because all proteins have everything and sometimes it's a mixture, it's not a unique property of any protein. हाँ ये ज़रूर है कि कोई दो प्रोटीन किसी जगह पर एक मास टू चार्ज रेशियो रख सकती हैं. सिमिलरली दो प्रोटीन्स एक मौके के ऊपर सिमिलर मास रख सकती हैं. कुछ प्रोटीन्स का सिमिलर चार्ज भी हो सकता है. सो देयर आर मल्टीपल प्रोटीन्स विच आर ऑफ द सेम कैटेगरी जिसकी हम बात कर रहे हैं, तो इसलिए इस तरह का बनाना मुश्किल है. हाँ ये तो ज़रूर हो सकता है कि यू मेक स्मॉलर चार्ट्स फॉर एग्जांपल साइटोप्लाज्मिक प्रोटीन्स के लिए एक चार्ट बन गया, या आप कहें कि एक प्रोटीन फैमिली के लिए एक चार्ट बन गया. लेट्स टॉक अबाउट फॉर एग्जांपल ट्यूमर सप्रेसर प्रोटीन्स. यू कैन मेक अ चार्ज ऑफ फॉर ट्यूमर सप्रेसर प्रोटीन, जैसा कि अक्सर होता भी है, लोग करते भी हैं कि 10 किलो डाल्टन की प्रोटीन है, 100 किलो डाल्टन की प्रोटीन है. सो दे डोंट केयर अबाउट एनीथिंग एल्स व्हेन दे थिंक ऑफ दोज प्रोटीन्स in a protein family. So you can make such protein charts for protein families, but not for 10,000 proteins which are present in the body, because by the end of the day it will be a Venn diagram. आपको पता है Venn diagram कैसी होती है? Venn diagrams are made up of circles. So you have one circle and then you have a circle which is encompassing this circle, and then there is a third circle जो कोई circle को भी, तो there are multiple things जिसको शायद हम कंट्रोल ना कर सके. मेरे ख्याल में मौसम बहुत खराब हो गया बाहर, है ना? सो ऐसा नहीं है कि हम इनका बना सके, कोई एक पीरियोडिक टेबल नहीं बना सकते. जी प्लीज.
थैंक यू सर.
जी, एनी फर्दर क्वेश्चंस? इफ नॉट वी विल मीट टुमारो. मैं आपकी